All right, welcome back. So today we're going to take a look at chapter 16 and 13. We kind of put them together because they'll make a little bit more sense, because really what we're talking about are the different pathways that go to and from your brain and how your brain interprets information. So it makes the most sense to really talk about the spinal cord here and these pathways. Okay, so with that, let's get started. I'll hide myself here. So just a little bit of background. So connective tissue covering. So if you remember back to muscles, right, remember how we had each individual muscle fiber was surrounded by connective tissue, and then we bundled those muscles together, and then we had connective tissue around that, and then we put all the bundles together, we put connective tissue around that. Remember we had endomysium, perimysium, and then endomysium, or epimysium rather. We have the same thing here, but with neurium. So take a look. So here's one entire spinal nerve, so as it kind of shows you here. So here's a spinal nerve. And you'll notice that individual axons get bundled, again, into these fascicles. Right? So here's a single axon. So around the axon, around that myelin sheath, okay, so you'd have the axon cell membrane, then you'd have the myelin sheath around that, and then around that, you're going to have endoneurium. Right? And then you bundle them together in a fascicle, and then we have connective tissue perineurium, keeping them together. And you're going to take several bundles and you're going to package them together into an entire spinal nerve, and then we cover that with epineurium. Okay, and then these are basically going to make up the dura mater. Will blend into the dura mater, or the dura mater will blend into the epineurium, either way, however you want to look at it. So basically, right at that intervertebral foramen, so right at between those two vertebrae, if you remember, make up that intervertebral foramen. That's basically like where this epineurium will blend into that dura mater. So hopefully that makes sense to you. We'll see something that looks like this in lab underneath the microscope. So what you're looking at here, so here's a big fascicle. So here's a big fascicle. There's about 3 in this image, right? One full one, another one over here, and then another one over here. Right, so around this big fascicle is perineurium. Epineurium will be around this whole thing. And then if we zoom in, what you see here labeled as A, these little dots are the axons. That white that you see around them, that's the myelin sheath. And then basically around that is going to be the epineurium. I'm sorry, the endoneurium rather. So the endoneurium around each individual fiber. All right. So right, these nerves are going to bring in sensory information to our brains. So the very first thing that's going to be really important that we discuss is what's the difference between something that's a sensation versus our perception. Okay. So here's our definitions. A sensation is going to be a conscious or subconscious awareness of changes in the external or internal environment. Right? So it's simply we understand that something has taken place. So for example, you turn on the lights in a room, you sense that it's become brighter, right? That's a sensation. You realize it's gotten lighter, right? Or you turn off the lights, it's gotten darker, right? Now, perception is going to be the conscious awareness and interpretation of those senses, right? So maybe you turn on the lights and you interpret that as a bad thing because it hurts your eyes. Or you turn off the lights and, oh, it's dark and now it's scary. That's your perception of that sensation, right? I mean, the lights on or off is neither good nor bad necessarily. It just is. However, that perception that you put on it, that interpretation, is going to be our perception, right? And a lot of times our senses are actually going to conflict one another. Sometimes our vision is going to conflict with what we hear, right? And your brain actually has to make sense of it. Like, which input is going to be more important than the other, right? Or sometimes vision versus touch will conflict with one another. And your brain has to make sense of that. And just like we talked about in the previous lectures about the brain, sometimes the brain's like, I don't get it, and it short circuits and it makes up its own thing. So take a look at these 2 videos down here at the bottom. Right? So just stop this recording and take a second. You've got these YouTube links. They are so much fun. I think you're even going to show your family. You're going to try this at home. So go ahead and watch these 2 videos about perception, and kind of just keep that in mind, and maybe we can talk about it in lab. All right, so I'll see you guys when you come back. All right, so hopefully You enjoyed those videos? I think the McGurk effect is just absolutely phenomenal. Like, I think it's kind of wild to imagine that basically the visual input overrides what we actually hear. It's just so weird. Like, and we don't even realize it, right? I mean, how natural was it that you heard those two different sounds? Or even more so that some like fake rubber arm, all of a sudden you think it's like your arm. That's crazy, right? When you really think about it, it's amazing how the brain just decides to do that, how it makes sense of the world. Anyway, I thought those videos were cool, so hopefully you guys enjoyed them as well. All right, so let's talk about some different types of sensory receptors. So as far as structurally, we have three different sort of structural configurations. We have what are known as free nerve endings, encapsulated nerve endings, and the ones that are actually separate cells. Okay. Come on, slide. All right. So the first one we're going to talk about are free nerve endings. And they're called free nerve endings because those dendrites are bare. So here you can see the dendrite, there's no covering on it or anything. It just is. It just ends there. Okay. So there are no specialized structures. So types of information that free nerve endings will convey are things like pain, temperature, tickle, itch, and then some touch. Okay. And we'll see it in a little bit that there's different nerve types. They're all free nerve endings. None of them have any kind of specialized structure. However, special ones will tell you information about pain. Other ones tell you about temperature. Okay, stuff like that. Although the same ones will tell you about tickle and itch and some touch as well. All right, I'll hide me here so you can see this better. The next sort of step is if we take that nerve, that dendrite, and then we wrap that in some sort of capsule. So these are encapsulated nerve endings. You saw these when we looked at the Pacinian corpuscle or Meissner's corpuscles. Those all had a capsule around them. Okay, that capsule is made up of different connective tissue. And then depending on the size of it, it'll determine the sensitivity and how specific that is. So we'll get different somatic and visceral sensations. So somatic is basically like touch, muscle sensations. So those and visceral sensations are things like anything that will relate to like your stomach and intestines and your internal organs are your visceral sensations. So you'll have like pain or I shouldn't say pain, but pressure, vibration, touch. It's like inside your actual organs themselves. So again, the example of an encapsulated nerve ending would be a Pacinian corpuscle. So here, there's some sort of pressure that would then bend that capsule, which then bends the dendrite, which then generates a potential, which will then trigger an action potential. And then that signal goes down the axon to the cell body and then onward to the central nervous system, to either the spinal cord or to the brain itself. And then we'll have some sensory receptors that are actually separate cells. So here we take a look. So like, these would be things like hearing, taste, sight. So I mean, think about it. Those senses are really, really super specific to like one thing, right? Taste is it's just taste. Sight is very specific. So we have actual specialized cells for that. Okay. So here's the example. Like right here. So here's a sugar molecule. So here's this special receptor. It's a cell all by itself. It picks up on that sugar molecule. It releases a neurotransmitter, which then stimulates the postsynaptic neuron, which then will, as long as it triggers an action potential, will send that signal down the axon, to the cell body and then to the central nervous system. Some other sensors that we'll talk about, or some other sensory receptors that we'll talk about. Okay, let me get my pen here, and actually just do that. Lost my pen. All right. So we have basically we can kind of categorize any of your receptors as either one of these two big umbrella categories. That's kind of why I drew this dashed line. So these two are just big overall categories. So the receptors are either going to tell you about information or about sensations or information of things at or near your body's surface. So they're telling you about your external environment. Interoreceptors will provide information about your internal environment. So it's one of those two. It's either telling you about stuff outside of you or inside of you. There is no other thing, right? It's two categories. Okay. And then so these are different types of either exteroreceptors or interoreceptors, or sometimes you'll find them both. Okay. So a proprioceptor is going to give information about movement and body position. Okay, so I think I mentioned this to you guys last time. Proprioceptors are going to be important because they're the ones that are allowing you to know that, oh, my arm is over here relative to my face. So when I go to take my glasses off, I'm not just smashing myself in the face, right? They'll also let different parts of your central nervous system, your brain, know that, oh, look, you've just got up and started moving. So they'll monitor that movement, like of your legs, of your arms, and they'll send that signal to the brain. The brain goes, oh, okay, well, we're active. Let's increase our heart rate. Let's increase our breathing, right? So we don't like run out of oxygen, for example. Okay. Mechanoreceptors are going to be receptors that respond to a physical stimulus. So things like touch, uh, stretching. So you'll have mechanoreceptors like at the surface of your skin. So sometimes those can be exteroreceptors. Sometimes you'll have mechanoreceptors like in your stomach, judging the stretch of your stomach. So, hey, stop eating now. Our stomach is maximally stretched. So in that case, it would be an interoreceptor. So mechanoreceptors are a really good example of where we can have some giving us information about the outside of our body, right? Someone touching you, versus like stretch going on like in your stomach because you've had a full meal. That'd be an interoreceptor. Thermoreceptor, I think, is fairly obvious, right? So these are going to give information about temperature, and we'll talk more about these in a second. Nociceptors are a specialized receptor that tell us about pain. So if you do not have nociceptors in your body, and there are some people that are born without them, I'll have you guys watch a video, they don't feel pain at all. None. Interestingly, and total aside, um, when you go fishing, right? You know how, well, hopefully you hook the fish in its mouth, right? Fish don't have any nociceptors in their mouths. So they feel you tugging, they feel the hook and they feel the tug, but it doesn't actually hurt. I know it's a weird thing to actually kind of think about, right? Like, how does it not hurt? They do not have this pain receptor. Again, we'll get to this. I'll have you guys watch a video, and hopefully that'll make a lot more sense and give you a better idea of what that actually looks like. All right, photoreceptors. This is basically vision. So photoreceptors are going to give us visual information. That's our sight. Chemoreceptors are going to give information about chemical cues. Okay, so typical chemoreceptors, classic ones, all right, so just easy to think about, are things like our sense of smell, sense of taste. Those are all chemoreceptors. They're picking up on some sort of chemical, right, in either our food or in the atmosphere or wherever. You'll have other chemoreceptors in your body as well that are picking up on things like pH. Let's see, just different chemicals that might be released by bacteria, even. So you'll have different chemoreceptors in your intestines that are picking up on like bad chemicals that maybe something you ate has all these bacteria. The bacteria releasing chemicals, and that'll let your intestines know, like, that they send that signal to the brain going, oh, no, you got to throw up, right? Or it's got to go out the other end because there's something bad in here, right? So anyway, so chemoreceptors give information about chemical cues. Osmoreceptors give information about hydration levels. Okay, so osmoreceptors are going to let your body know, hey, this person's dehydrated, there's not enough water, or there's tons of water, you need to produce more urine, something like that. Okay. All right. Some of these receptors are going to be what we call rather rapidly or slowly adapting. And basically, what that means is that these are not going to always fire continuously. Okay, usually these receptors are going to adapt to whatever the stimulus is. All right. So most sensory receptors exhibit adaptation. So that's the tendency for the generator or receptor potential to decrease in amplitude during a maintained constant stimulus. Okay. So, and I'll explain that more. I think the next couple of slides it'll become a little bit more obvious. And I think these pictures, so this is like rapidly, the roadrunner is rapidly adapting, and a tortoise is slowly adapting. And I just thought this was really cute. It's a abandoned baby hippo that made friends with this big old Galapagos tortoise. I just thought that was adorable. So anyway, that's what those images are. All right, so here we go. Adaptation of sensory receptors. So again, rapidly adapting. So these are things like your sense of smell, pressure, touch. So for example, and you probably all know this, right? Let's say you go into some place and it has like a funny smell, right? After a while, you don't smell it anymore. Probably, I don't know. I don't know if you guys have ever worked at a restaurant or someplace that handles food. And I mean, you've worked in it all day, so you don't really smell it anymore. You go home and the person meets you and you're like, oh, you smell like, I don't know, fryer grease. Go take a shower, change your so you're like, really? I smell it? I can't smell it. Or if you've been camping, right? You don't smell yourself after a while, mercifully so. So again, your smell adapts actually fairly rapidly, actually. It doesn't take very long. After a few minutes, you've pretty much adapted to the smell. Same thing is true for pressure and touch. I mean, think about it. I mean, when you first put like your socks on, you realize, okay, I put socks on. But you don't go like all day, like, oh, I have socks on my feet. My feet have socks. Oh my God. Like you don't keep worrying about it because your receptors have adapted to it, right? You probably don't even pay attention to like the clothes that you're wearing unless they're uncomfortable or itchy or something, right? So these sorts of rapidly adapting receptors, these rapidly adapting receptors are specialized for detecting changes. Yes, the smell is here. I recognize it. And then I don't care anymore. It'll adapt. And then once the smell is gone, you might notice that, right? Same thing with putting your clothes on. Yes, I have clothes on. I realize that. I have no clothes on now. I realize that. And after a while, you get used to that as well. Unless it's like a really awkward situation, which maybe not, but whatever. Then slowly adapting receptors are exactly the opposite, right? So that signal gets sent the entire time the stimulus persists. Okay. So for example, pain, right? If you have pain, it's not like after, you cut yourself, let's say it's a pretty decent sized cut, it's not like the pain goes away after a minute or two, right? It's there for a really long time. You don't want to ignore pain because it implies there's some sort of structural damage. So let's say you really damaged your leg, I don't know, broke your tibia or something, it hurts and it's going to keep on hurting until it's fixed because it's basically telling you like, hey, don't stand on this thing, it's broken, right? Don't try to run, you're not going to get away, or you're going to have to figure something else out. Same thing about body position, right? I mean, all of us, if we're sitting here, you're laying wherever watching this video, right? It's not like you always want to go like, whoa, where am I sitting? Am I standing? What am I doing? Where's my head? Right? You remember what your body position is, right? So again, those are slowly adapting receptors. You don't want to ignore these kinds of information. They're really important. All right. So when we take a look at this, so for example, this is what a slowly adapting receptor would look like. So here in green is the stimulus. So again, this is sort of just a science textbook kind of diagram. So here's a stimulus. It's nothing's happening, and then we turn the stimulus on. We maintain a constant stimulus, and then we turn it off, right? And in the pink line here is what the firing of the actual axon, so a slowly adapting receptor. So again, nothing, turn the stimulus on. It takes hopefully you can kind of see this, a little bit of a lag time. It responds, and for the most part, it continues to respond the entire time. And then the stimulus is gone, it stops. Now, if we take a look at what a phasic receptor, so this is a rapidly adapting receptor. So again, the green line is our stimulus, so nothing. We turn the stimulus on, whatever that might be, turn it off. So here again, now we take a look at the receptor in pink. Nothing, nothing, nothing. We turn the stimulus on. There's a little bit of lag time. Receptor goes, oh, hey, I smell it. I don't care anymore. It stops firing. And then even though we have the stimulus, it's not firing anymore until we turn the stimulus off, and then it goes, hey, there was a change. Okay. So again, what this is important for is monitoring changes, right? So this one, not so much. This is important for that information that so the slowly adapting, important for that information that you have to always remember that you have, again, pain because of structural damage. So you don't want to forget that. All right. I'm actually going to stop the video there because that's a good stopping point. So let me know if you have any questions, and I will see you in the next video.